Good evening, everyone. Today, our group is going to talk about the molecular pathogenesis and screening program of colorectal cancer. We will first introduce the background information of colorectal cancer. Then we will discuss the three molecular pathogenesis pathway and their molecular tests. This pathway, including chromosomal instability, microsatellite instability, and CpG island methylation. And finally, we will talk about the screening program in Hong Kong. Colorectal cancer is the neoplasia of the epithelial linings on colon and rectum. This is the third most common cancer in Hong Kong with a high incident rate. And this is also the second leading cause of cancer death in Hong Kong, with a 5-year survival rate around 16%. And due to the late onset of the symptoms, around 16% of the new cases are diagnosed as metastatic, with limited treatment decisions. Therefore, launching our screening program can help to screen out the colorectal case at the early stage and provide suitable treatment, and finally can lower the mortality. There are several factors that can higher the risk of getting colorectal cancer, including aging, lifestyle, obesity, and smoking. Besides, having family history of colorectal cancer and history of inflammatory bowel disease or Crohn's disease may also higher the risk of getting colorectal cancer. There are 3 types of mutation in colorectal cancer, including sporadic, inherited, and familial mutation. Sporadic colorectal cancer accounts for more cases and mainly point mutations. Inherited colorectal cancer are related to the inherited gene and can be subdivided into familial adenomatous polyposis and hereditary non-polyposis colorectal cancer. And for familial colorectal cancer, it is also related to the inherited gene. However, their genetic composition and pathogenic pathway are not well defined. The colorectal cancer usually takes decades for the cancer progression from normal epithelial cells to hyperplastic lesion, from adenoma to carcinoma, and finally to the metastatic carcinoma. The whole development usually involves at least 2 to 3 mutated driver genes. There are 3 molecular pathogenesis leading to colorectal cancer, including chromosomal instability, microsatellite instability, and CpG island methylation. I will first talk about the chromosomal instability. Chromosomal instability accounts for most colorectal cancer and can define it as imbalanced number of chromosomes, results in loss of heterozygosity. Multiple driver genes are involved in the cancer progression, and they can be defined into 3 categories, such as first driver genes, second driver genes, and third driver genes. For the first driver genes, such as APC genes, mutation in these genes can result in hyperplasia of the colonic epithelial cells and promote the risk of a second driver gene. Second driver genes, such as KRAS and BRAF, the mutation of these genes can contribute to out-of-control proliferation and the formation of adenoma. The third driver genes, such as SMAD4, TP53, and PIK3CA gene, the mutation of these genes can allow the tumor cells to invade the surrounding tissue and become metastatic. And I will discuss these driver genes one by one. First, let's talk about the first driver genes. APC genes is the most prevalent driver genes at the early stage, which is found in more than 30% of colorectal cancer patients. It is normally involved in the WNT pathway to regulate cell proliferation, cell-cell adhesion, and cellular integrity. Mutation of the APC genes can produce truncated APC proteins and cause the constitutive signaling of the WNT pathways, and finally leads to the hyperplasia of the colonic epithelial linings. Next, for the second driver genes, KRAS and BRAF are the most common genes. KRAS activates the EGFR and initiates the two signaling pathway to regulate the cell proliferation and apoptosis. Mutated KRAS produce defective KRAS proteins, which can activate the signaling pathway without EGFR activation and cause the cell proliferation and finally the adenoma formation. BRAF gene is also involved in regulating cell differentiation, proliferation, and apoptosis. Mutation on the BRAF genes also contributes to the adenoma formation. And finally, for the third driver gene, SMAD genes involved in the TGF-beta pathway, while the TP53 genes play roles in the cell cycle checkpoint. Both genes can control the cell proliferation. Mutation on these genes not only cause the uncontrolled cell growth, but also increase the risk of epithelial mesenchymal transition. And this makes the cancer cell prompt to metastasis.
For the second molecular pathway of colorectal cancer, I'm going to introduce microsatellite instability, MSI. It contributes about 15% of all colorectal cancer cases, in which less than 25% are hereditary cases, and the remaining cases are sporadic, which will be introduced by my group mates later. Now I'm going to focus on the hereditary cases. Before understanding what is MSI, we have to know what are microsatellites. Microsatellites are the repetitive short tandem repeats, so they are highly prone to replication errors due to polymerase slippage. MSI is a hypermutable phenotype which are resulted from accumulated insertions or deletions in the sequences because of the deficient mismatch repair system. Normally, DNA mismatch repair system MMR maintains genomic integrity by detecting and correcting base space mismatches and insertion deletion loops during DNA synthesis. It involves key heterodimeric complexes, which are mut S alpha containing MSH2 and MSH6 for single base mismatches and small loops. Also, mut S beta containing MSH2 and MSH3 for larger loops, and the mut L alpha containing MLH1 and PMS2. They act as the endonuclease. Upon mismatch recognition by mut S alpha and mut S beta, followed by the recruitment of mut L alpha, it initiates the excision of the erroneous strand by isonuclease 1. And then the DNA polymerase delta utilizes the parental strand as a template to resynthesize the gap, and the gap is sealed by ligation. Therefore, defects in any major MMR proteins lead to the mismatch repair deficient and increasing the mutation rate about 100 to 1,000-fold compared to MMR proficient cells. This mutator phenotype leads to widespread alteration in microsatellite repeats across the genome, causing MSI. For the hereditary cases of MSI, see colorectal cancer. In Lynch syndrome, which also called hereditary non-polyposis colorectal cancer, germline heterozygous pathogenic variants in MMR genes, most commonly in MLH1 and MSH2, are inherited in an autosomal dominant pattern. Therefore, a somatic second hit that inactivates the remaining wild-type allele will cause biallelic inactivation and then dMMR, causing high MSI and lead to tumor development. Lynch-associated MSI high colorectal cancer typically occurs at age of average 45 to 50, and the tumors are frequently located in the proximal colon and presents with synchronous and metachronous tumor. These tumors commonly show poor differentiation, mucinous histology, and lack BRAF V600E mutation. The Down syndrome consequences of dMMR-driven MSI are essential in colorectal carcinogenesis. Unrepaired replication errors preferentially target coding microsatellites in tumor suppressor and regulatory genes. Typical targets the genes include B8X, which is the pro-apoptotic regulator, and transforming growth factor beta receptor type 2, also the RNF43, which is the negative regulator of the WNT signaling pathway. These mutations disrupt critical pathways like evasion of apoptosis and promoting the uncontrolled cell division and excessive cell proliferation. This significantly increased the tumor mutational burdens, also produced a large number of neoantigens from frameshift mutations, triggering a strong anti-tumor immune response like a large amount of CD8 positive tumor infiltrating lymphocytes and an immunological hot tumor microenvironment. This robust immunogenicity is the hallmark of the MSI high tumors.
The third molecular pathogenesis is CpG island methylator phenotype, CIMP. CpG islands, GCI, are short GC-rich DNA sequences with high CpG dinucleotide frequency. Normally, promoter CGIs are unmethylated, enabling transcription and expression of tumor suppressor gene to regulate cell growth and apoptosis. CIMP accounting for approximately 20% of sporadic CRCs. It is epigenetic modification involving extensive methylation of 5' cytosine residues within the promoter regions, causing transcriptional silencing of tumor suppressor gene. This disrupts the cell growth control and promoting oncogenesis. The exact CIMP mechanisms remain unclear, but evidence shows that they may involve DNA methyltransferase overexpression, mutation of genes responsible for chromatin remodeling, and histone changes. Environmental factors like smoking and diet may also promote the normal colon cells to this phenotype. CIMP is associated with BRAF mutation, with BRAF V600E being the most common mutation type, accounts for 90%. BRAF mutation is caused by the T to A transversion and leads to valine to glutamate substitution. BRAF mutation causes uncontrolled proliferation and apoptosis failure. BRAF mutations followed by CIMP drives serrated polyp pathway. A hyperplastic polyp is transformed to cystized serrated adenoma and then proximal colon cancer, often with poor prognosis. CIMP also links to MSI through MLH1 promoter hypermethylation. It allows transcriptional silencing of MLH1 gene and fails to be expressed. Under mismatch repair deficiency, errors in microsatellites during replication fails to be repaired. Replication errors accumulate and lead to MSI-mediated oncogenesis, which regarded as sporadic MSI high tumor. This helps distinguish them from Lynch syndrome, and it highlights interrelated CLC molecular pathways.
Colorectal cancer can be diagnosed by molecular tests. For the mutation due to chromosomal instability, next-generation sequencing and digital PCR are highly recommended. For NGS, iron tolerant and Illumina are the commonly used NGS platforms for detecting the hotspot mutation of colorectal cancer. The iron amplified sequence colon panel from iron tolerance and the next sequencing 500 panel from Illumina claimed to detect 22 and 73 genes targets respectively, including KRAS, BRAF, TP53, PIK3CA, SMAB4, etc. And for the digital PCR, Pico droplet digital PCR can detect KRAS and BRAF.
For the detection of MSI mutation in colorectal cancer, there are three main routine tests. First and most commonly used is immunohistochemistry. It tests for the loss of MMR protein. Loss of nuclear staining indicates the MMR deficiency and strongly correlates with high MSI. It is fast, cheap, and widely available test. Secondly is PCR-based MSI testing, which remains the gold standard for confirmation. A more sensitive pentaplex mononucleotide panel is more preferred than the original Bethesda panel because it offers higher accuracy and better performance. Thirdly, next-generation sequencing is increasingly adopted. It can simultaneously detect more detailed mutations informations in one test, although it is more expensive. Emerging methods include artificial intelligence and circulating DNA-based testing.
PCR-based molecular techniques are used for detecting promoter hypermethylation of CIMP. Methylite is quantitative PCR to measure methylation by calculating percentage of the methylated reference, which offers high sensitivity and throughput. Methylation-specific PCR is qualitative assay by using two primer pairs that detect the methylated and unmethylated DNA. Methylation-specific multiplex ligation-dependent probe amplification quantified DNA methylation status and copy number for multiple targets in one reaction. Unlike methylite and MSP, sodium bisulfite pretreatment on DNA is not required for MSMLPA. Whole genome bisulfite sequencing is single-base resolution methylation mapping across the entire genome, enabling comprehensive epigenomic profiling. CIMP classification lack of standard, but key panels include Weisenbergen's 5 markers panel includes CANA1G, IGF2, NeuroG1, runs 3, SOCS1, more than or equal to 3 of the 5 markers methylated classifies as CIMP positive. Original extended 8 marker panels at CDKN2A, CRABP1, and MLH1 to the above 5 markers. More than or equal to 6 out of 8 markers are regarded as CIMP high. This correlates with BRAF and KRAS status for subtyping. Further sensitivity analysis on markers panels are recommended to verify the CIMP subtyping.
Let's move on to talk about the screening program of CRC. The framework for CRC screening program is illustrated here. First of all, it is recommended for all adults aged 45 to 75 to participate in the program, while older adults aged 76 to 85 should be determined by their overall health. There's a difference between preventive screening and early detection screening. Preventive screening is for the patients that before CRC actually occurs, while early detection screening allows early detection when the screening is taking place at an early stage of CRC development. So, moreover, we have to also consider the benefits and harm of the screening program. The benefits include reducing CRC incidence and mortality, while the harm is that if overdiagnosis occur, it may cause anxiety to the patients. So the picture here shows the framework for screening program, where a pre-screening fecal testing or preliminary endoscopy are taken. Positive results from these tests will refer the patients to a detailed colonoscopy, which classify them into low risk, high risk, or CRC diagnosed cases. Those diagnosed with CRC will receive treatment immediately, while those with a risk for CRC development in the future should undergo surveillance in a certain period of time. These are the major CRC screening methods and their principles. For the traditional guaiac-based fecal occult blood test, it works by testing the peroxidase activity of hemoglobin heme group, while for the more accurate fecal immunochemical test, it works by antibody binding, with a positivity defined by fecal hemoglobin concentration above 10 microgram per gram of feces. For colonoscopy and sigmoidoscopy, these are endoscopic methods, while sigmoidoscopy restricts the scope to the sigmoid colon only. There are also alternative methods, including direct visualization by capsule endoscopy, CT colonography, and also stool-based multi-target stool DNA. Non-invasive methods by testing CRC biomarkers in circulating cell-free DNA has also been developed. In order to select the best method for screening, we have to consider a few factors. For FOBT, dietary restrictions include the patient should prevent intake of reducing or oxidizing agents from their diet, which results in false positivity or negativity. And also through stool samples are required for this test. While FIT, it can be done with a single stool sample only. For colonoscopy, it has a higher risk of complications because of the need of bowel preparation. And also it is a more invasive procedure that may cause bleeding or bowel or colonic perforation, while sigmoidoscopy is relatively safer because it restricts the scope of endoscopic examination. Meanwhile, it may has a chance of missing diagnosis, and the need of a second colonoscopy follow-up if any abnormal results may also increase the overall risk of the patients. So these are the additional consideration while comparing the different screening methods. We can see that for FGOBT and FIT, they are more cheaper methods. But meanwhile, their sensitivity or specificity is relatively lower. For colonoscopy, it is the gold standard for CRC diagnosis. The need for preparation and the risk of complications mentioned above make it tests that should not be performed so frequently. For sigmoidoscopy and cfDNA, there are also different concerns regarding their location and also sensitivity for polyps. So for CRC screening program in Hong Kong, the government also recommend individuals aged 50 to 75 to perform FOBT tests twice every 2 years. In case any positive case figure out, they are referred to colonoscopy. And these are for the general population. For population with a higher risk of CRC development, detected by positivity of mutations for Lynch syndrome, familial adenomatous polyposis, and also having a first-degree relative diagnosed with CRC. For these group of people, they are recommended to take the colonoscopy in a shorter interval, which is 5 years, starting from 40. So recently, the screening program in Hong Kong has already covered around 40% of the target population, while a higher percentage is anticipated to be covered in the future in order to achieve early CRC diagnosis. In conclusion of this part, we should know about the cost-benefit analysis of different screening programs in order to choose the best method. In the future, the discovery of new biomarkers for non-invasive CRC screening, the incorporation of AI-assisted colonoscopy, and the development of practical guidelines and regulations are also included. In order to enhance the service quality of screening program, ethical concerns by a thorough informed consent and quality assurance should also be included.